Module 8. Genetic Evaluation, Selection, and Breeding Strategies in Livestock. Module Overview. Module 8 provides an in-depth exploration of genetic evaluation and breeding strategies in livestock production, emphasizing quantitative and qualitative traits, selection methods, and breeding program design. The module integrates classical genetics, quantitative genetics, statistical tools, and modern genomic approaches to provide a comprehensive understanding of how to assess, select, and propagate superior genetics effectively. The module addresses how systematic evaluation of animals, combined with strategic mating plans, can maximize genetic gain, improve productivity, enhance disease resistance, and optimize economic efficiency. It also examines crossbreeding, linebreeding, inbreeding, and hybrid vigor, heterosis, alongside the practical implementation of breeding objectives in commercial and research settings. Key concepts in genetic evaluation and selection: genetic parameters and quantitative traits, understanding heritability, genetic correlations and breeding values for traits such as growth, milk production, fertility, and disease resistance. Differentiation between qualitative, single-gene traits and quantitative, polygenic traits. Use of statistical models, BLUP, Best Linear Unbiased Prediction, REML, restricted maximum likelihood for accurate genetic evaluation. Selection methods, individual selection, choosing animals based on their own performance. Family or pedigree. Selection, using the performance of relatives to select breeding candidates. Progeny testing, evaluating sires or dams based on the performance of offspring. Genomic selection, utilizing DNA markers and genomic information to accelerate genetic improvement. Breeding strategies. Pure breeding, mating within a breed to maintain or enhance breed characteristics. Crossbreeding, mating between different breeds to exploit heterosis. Hybrid vigor, for improved productivity. Linebreeding, mating of related animals to concentrate desirable traits while minimizing inbreeding risks. Inbreeding, used cautiously to fix traits but requires careful management to avoid inbreeding depression. Selection intensity and accuracy. Selection intensity, proportion of individuals selected. Higher intensity increases genetic gain but may reduce genetic diversity. Selection accuracy, reliability of predicted breeding value, affected by data quality, pedigree information, and trait heritability. Genetic gain and response to selection. Genetic gain, delta G, improvement per generation, calculated using heritability, selection differential, and generation interval. Strategies to maximize delta G. Shorter generation intervals, accurate evaluation, and selection of high-performing animals. Economic and breeding objectives. Defining breeding goals, productivity, reproductive efficiency, disease resistance, environmental adaptability, and market traits. Economic selection indices combine multiple traits with weighted economic importance to rank breeding candidates. Use of technology in genetic evaluation integration of computerized breeding records, performance testing, and genomic tools. Application of marker-assisted selection, MAS, and genomic selection to accelerate improvement of complex traits. Monitoring genetic diversity using molecular markers to avoid genetic bottlenecks. Practical applications designing breeding programs for dairy, beef, sheep, goat, and pig operations, using selection indices and reproductive technologies. Using crossbreeding strategies to exploit hybrid vigor and improve production efficiency. Applying genomic selection to reduce generation intervals and improve response to selection. Developing conservation strategies for endangered livestock breeds while maintaining genetic integrity. Evaluating economic impact of selection decisions on herd productivity, profitability, and sustainability. Learning outcomes. By the end of Module 8, you will be able to understand and calculate key genetic parameters, including heritability and breeding values. Apply appropriate selection methods for various production traits. Design and implement breeding strategies to maximize genetic gain and efficiency. Integrate genomic information into selection and breeding programs. Evaluate the economic implications of genetic improvement and breeding decisions. Monitor genetic diversity and mitigate risks of inbreeding. Develop practical breeding plans for commercial, research, and conservation livestock operations.